Okay, folks. Today we are going to learn about amino acids. So hopefully you've been following along on Top Hat, where I've asked you to kind of start memorizing the amino acids section by section. For the learning check for amino acids, I will expect that you know the name, structure, and abbreviations for all of the amino acids. So hopefully you have good time management and you've been working up towards memorizing them all over the last couple of weeks. Other learning outcomes: recognize the different pKa's of amino acids, classify amino acids according to their R group, draw individual amino acids as well as polymeric peptides, and understand how the environment of an amino acid can affect its pKr. Today's Meet a Scientist moment is William Rose. He was a professor at University of Illinois Urbana-Champaign, and he was able to determine that some amino acids were essential in the diet, therefore essential amino acids. He also discovered threonine. Now, his research happened a long time ago and wasn't exactly ethical according to today's standards. For example, he did experiments on his own graduate students. So how did he know that an amino acid was essential in the diet? Well, he'd feed them very specific, restricted diets that didn't contain amino acids, and he would see if they got sick or not. Would not recommend. Let's get into it for today. What are the amino acids we have to know? Well, there are 20 common alpha amino acids. We'll just refer to them as amino acids. Most have a primary amino group. So you'll see here a primary amino group, an amine that is bonded to a single carbon. The only exception to this rule is proline, pictured below. It is the only amino acid that is secondary. You can see it is bonded to 2 carbons. What should we understand about the structure of amino acids? Okay, well, we have what we call the backbone of the amino acid. This is common between all amino acids. We have the N-terminus. We have the alpha carbon and the C-terminus. Actually, let me just undo that real quick to show you first just how it looks like with its proton. We'll talk more about that later. The N-terminus refers to the amino terminus. The C-terminus refers to the carboxylic acid. And that is the backbone. The N, the alpha carbon, the COOH. Every amino acid also has an R group, and the R group is what makes each amino acid different. So really, when we talk about amino acids, we focus in on that R group quite a bit. What about the pKa's of amino acids? Well, some amino acids have 2 pKa's, some amino acids have 3. Every amino acid will have a pKa for its C-terminus. That pKa for an amino acid is about 2, but when we plop that amino acid into a protein, it's about 3 to 4. Every amino acid also has a pKa for the N-terminus. That amino pKa in an amino acid, closer to 9, but once we plop it into a protein, it's closer to 7 to 8. Now, some amino acids have R groups with pKa's, and these are kind of diverse depending on what the identity of the functional group is. At this point, folks, I'll point you to a handout if you haven't already seen it on Top Hat. This handout is called Amino Acid Handout. There I have drawn and listed each amino acid with its pKa's. The pKa's are absolutely not something that I ever want you to memorize. Now, because we have so many pKa's, we should realize that amino acids can gain or lose protons at least 2 locations, sometimes 3. At physiological pH 7.4, amino acids are Zwitterions. They are dipolar ions. I will show you what this means. And I am showing you folks because I want us to understand that we should really be drawing these amino acids in the class as they appear in our bodies. So at physiological pH. And this relates back to the buffers material because we need to understand when a functional group is protonated or deprotonated. Let's start with the carboxylic acid group. The carboxylic acid group has a pKa of about 2. Physiological pH is about 7. pH is less than pKa. Oops, excuse me. Mom brain really got the better of me there. If the pH is 7.4, pH 7.4, pKa is 2. pH is larger than pKa. Do excuse me. If the pH is more basic than the pKa, more basic, that proton will have been lost. So if I draw my amino acid, C-terminus should have lost its proton since we are at a pH more basic than the pKa. What about that N-terminus? pKa is 9.4. pH is 7.4. So 7 is smaller than 9. If the pH is more acidic than the pKa, more acidic, more acid, we would expect the N-terminus to be protonated. And let's just leave the R group as is for now. So when I say that at physiological pH, amino acids are Zwitterions or dipolar ions, that is because the N-terminus is protonated and positive, and the C-terminus is deprotonated and negative at physiological pH. And so let's keep this in mind when we talk about amino acids, when we talk eventually about proteins as well. The C-terminus generally deprotonated, the N-terminus generally protonated, and the R group will really depend upon the identity of that R group.